Okay, so the story behind this movie is that in 2004, I was an adjunct professor at the University of Minnesota. And I went to a conference that was put on by NIDA, which is the National Institute on Drug Abuse in the States, one of the National Institutes of Health, NIDA. And they were staging a conference by a very odd name. It was called Potential Therapeutic Uses of Illicit Drugs. And I was invited to go to this conference, and I went. I presented my usual shtick about ayahuasca, and and then went home. And then 2 or 3 weeks later, I was contacted by one of the people that was in the audience, this fellow from a foundation called the Stanley Medical Research Foundation. And he wrote me an email. He said, "I saw your presentation, liked what you said," and so on. And he said, we support schizophrenia research, and we're interested in finding better medicines to treat schizophrenia, and particularly certain subset of symptoms called negative symptoms of schizophrenia, which are more like cognitive dysfunctions rather than drugs for psychosis. And he said, we're interested in natural products, and we have a program going on in China looking at Chinese medicines. But he said, we don't have anything in South America going, so would you be potentially interested in applying for a grant? And, I said, well, yeah, maybe. And, I wasn't really looking to apply for a grant, but he sort of made this offer. And he said, oh, by the way, we spent $170 million last year in grant funding for schizophrenia research. And basically I said, where do I sign, right? And I didn't get $100 million. I did get $500,000, which gave me the ability to go to Peru and start collecting plants that might be useful for actually dementias and cognitive deficits as much as schizophrenia, because there's an overlap. So I wrote up this proposal and submitted it to them and got the grant. So I went there first in 2004, and from 2004 to 2008, I was collecting regularly. And then 2005, I believe it was, this was all out of Iquitos. If many of you know, Iquitos is the kind of epicenter of ayahuasca tourism. But I'd worked out of Iquitos since 1981, and there's a botanist at the university there that I love to work with. So I went to Iquitos, and I had this grant funding. And in 2005, I was also there for the International Conference on Shamanism, which was organized by a local expat, Alan Shoemaker, who may be known to some of you. And so I was at this conference, and there at the conference, there was this young woman, Sarah Hutt, and she was there for the conference. And she was wanting to make a movie about traditional medicine. She was a freelance filmmaker. So she was there with her cameraman and so on. And I told her what I was doing. So we kind of agreed. She said, well, I'd like to follow you, and do some filming. So I said, well, sure, why not? I said, it's actually not that exciting, but if you want to come, you can come. And so she did, and she made this film, which at the time, I think it was, well, it's now called The Shaman and the Scientist. It was called In Search of a Cure or something like that at that time. So she followed me and put together this documentary. And then what goes around comes around. Some year, actually last February, this year, she invited me to the Philip K. Dick Film Festival in New York City, because she'd been invited to give this trailer. This is essentially a 20-minute trailer. She'd been invited to give it, and I thought, what? Why the Philip K. Dick Festival? This does not seem like an appropriate venue. But anyway, she had a friend who'd organized this festival. So they invited us, and we went. we showed this movie and we had a panel discussion, and what do you know, she got an award for the best short documentary at the festival, even though it wasn't science fiction and it wasn't even really that weird, you know. And so, but it was a very nice little film, and I was glad to see that she got this award. So we're actually going on the circuit again. I've got another conference coming up next weekend where she's going to join me in New Orleans, and we're going to show it again. But anyway, so this is the film that we'll show you. And I guess that's enough of an introduction.
Donde vayas, donde estés, por el camino que tú vienes, por el camino que vas, sirenita, encantando amorito. Sacarle a esto es cortarle, ¿no? Y uno se agrega acá un envase, se lo prende acá, entonces se va juntándose la leche. Es una medicina para purgante, ¿no? Y también para parásitos, todo tipo de enfermedad, ¿no? Es grandes doctores de la ciencia vegetal, espiritualmente, tienen espíritu, son vivos.
Nature is a much better chemist than chemists are. Plants have been synthesizing compounds for, 250 million years or longer. They've created a tremendous unexplored repository of molecular structures that are far beyond anything any chemist could ever dream up. There we go. Part one of the story, or really maybe I should say part two, because what I'm going to talk about now is how did I get into this? This is not a technical presentation. This is more or less a personal story of what led me to get into this sort of dodgy profession of ethnopharmacology. I just wanted to, as a follow-up to the presentation, I just wanted to show you, this is Sarah, the filmmaker. We're in our full native regalia here, as you can see. This Bora headband, we're in a village called Pucurquillo, slightly up near a town called Pebas on the Amazon, up a river called the Rio Ampiacu, which means the river of poisons. And I didn't go in for the face paint. And then this was our guide, the gentleman on the right there, Aku, or Richard Fowler. Quite a character, ex-special ops, ex-CIA, ex-whatever. I mean, he's amazing jungle guide. When I first met him, he said, this is what I do. I take people into the jungle to places they've never imagined exist. And he says, just give me the coordinates and I'll put you there. Well, it wasn't quite so difficult. Actually, it was pretty easy for us to get to this place. But this is the tail end of that expedition. So that was in 2006. So flash forward to 2017. And for many years, I've wanted to present this symposium, the Ethnopharmocologic Search for Psychoactive Drugs. And the reason I wanted to do this conference, it was kind of one of those bucket list items for me, was because in 1967, there was a very significant conference that nobody paid attention to. It happened in San Francisco, and in 1967, it was sponsored by the U.S. government, actually the National Institutes of Mental Health. And the government was later became quite embarrassed that they ever funded anything like this. It was a closed conference. Most of the people were well known in the field of ethnopharmacology, which was known to practically nobody. And they had this private conference, and they published a book called The Ethnopharmacologic Search for Psychoactive Drugs. And the book was very influential in my career when it fell into my hands somehow at age 18. And ever since then, I've wanted to do a follow-up conference. The government was supposed to do follow-up conferences about every 10 years. Well, it never happened, and they probably got a lot of flack because they'd sponsored this conference, about half of which was about psychedelics. But psychedelics weren't quite so stigmatized at that time. So they did that conference, and then there was never any follow-up. And I wanted to do one ever since. I wanted to do one for the 30th anniversary. Well, circumstances, funding, and good luck never really fell together. So 2017 rolled around, 50th anniversary of the first one, and it all came together. We had a beautiful venue to do it in the UK, this place called Tyringham Hall, where we were able to get some support through the Hefter Research Institute and the Institute for Ecotechnics, as well. And we were able to put on this conference, except this time it wasn't private. It was a very small number of people attended, but we did Facebook live streaming of all the lectures, which you can watch online. And I think if you've got a flyer, I think there's a link there, certainly a link on the website. At certain times, 60,000 people were tuning in and watching these lectures, which I thought was pretty amazing. A lot more people know what ethnopharmacology means now. At least it means that. So we did this conference, and one of the things I wanted to do with this conference, we invited Dr. Stephen Zara, who at 93 was not able to travel to the conference. But he submitted a very nice sort of video introduction welcoming us. And the reason that I wanted to invite Dr. Zara, he was for a time the director of NIDA back in the 50s. But before that, he was a Hungarian researcher in Budapest, and he wanted to work with LSD. But because he was behind the Iron Curtain, Sandoz refused to send him a supply of LSD. He was a psychiatrist, a pharmacologist, and a chemist, and basically he couldn't get a hold of LSD. So he decided to synthesize DMT. And he was the first person to definitively establish that DMT was a psychedelic. Doing it the only legitimate way you can, he injected it into himself. And he published an important paper called Dimethyltryptamine, Its Metabolism in Man, the Relation of its Psychotic Effect to the Serotonin Metabolism. So Dr. Zara was this rather amazing figure, an immigrant to the United States that became, the head of NIDA and had many other positions in NIH in that structure, but also a true psychonaut, a true explorer who established that DMT was a psychedelic. And he presented at the 1967 conference, and I was hoping to get someone from that conference to come to kind of bridge the gap. And he said, no, I'm too old, I can't travel. But interestingly, when he went to the conference, when he went to the 67 conference, he asked his so they gave the conference, and this was 67, Summer in Love was in full swing in Haight-Ashbury and all that. And Stephen asked his supervisor, so what's with these hippies, you know, and what's with the use of, what are they doing with these psychedelics? And his supervisor said, well, you know, why don't you go spend some time in Haight-Ashbury and see what's going on? And so he did. He went to Haight-Ashbury and spent a couple of weeks hanging out with the hippies. And he wrote this paper, like he wrote up the report, being like a good NIH scientist, right? He wrote up a report, a scientist among the hippies. And he was hoping this would be published, but his supervisor read it and said, there's no way that we're going to publish this paper. You know, you could just forget it. We're not going to encourage this kind of foolishness. And so he never published it. But when it came around to doing this conference, he said, well, I have this paper in the drawer that hasn't been published for 50 years. So I'll be happy to submit that to this anthology. So that paper in this book is published for the first time. Very interesting paper, you know, 50 years perspective on this. And so that's one of many goodies in that book. And so we dedicated the whole conference to him. I mean, this man was a pioneer and an amazing person. Well, to get back to the personal side of it, in 1968, the year that this conference, the year after the original conference was held, I was 17 years old. I was living in this small town in Colorado, Paonia, Colorado, bored to death, and hanging out with my friends, a couple of them here. We spent a lot of time drinking beer and singing protest songs and smoking a little pot, and generally, well, like teenagers everywhere, kind of an aimless existence. But we were very interested in psychedelics and the whole hippie thing that was going on in San Francisco. We had a hard time. We didn't really have access to psychedelics, even though my brother was out in Berkeley at this point. But this is where we wanted to be. We wanted to be where the action was. You know, we wanted to be at one of these human beings, and but it was aspirational, you know. And so we just persisted, and we're sort of marking the time when we could get out of Paonia and get on to the rest of our lives. And it was, you know, well, eventually we did. I mean, Paonia, for all its faults, was actually a very sweet little town, still is a sweet little town. But at that time, I only wanted one thing from Paonia, and that was out. So eventually I did make it out. And but around this time, and I don't know from what source, how this came into my hands, but the first edition of the Ethnopharmocologic Search for Psychoactive Drugs came into my hands. And about the same time, the first edition of the Teachings of Don Juan by Carlos Castaneda came into my hands. And I know exactly where that one came from because my brother gave it to me for my 18th birthday. Where this one came, I'm not sure, but I devoured both of them pretty much cover to cover. And it was an interesting juxtaposition because the Teachings of Don Juan, even though we now know that most of it was made up, or much of it was made up, it didn't really matter because what it gave me was a perspective that there was an ethnographic side to this and a whole anthropological aspect of psychedelic drugs. And at the time, you know, the hippies, that whole movement, they didn't really acknowledge that there was indigenous traditions. Everyone thought it all started with Timothy Leary, and not at all. So this made it clear, you know, that there was a wider story to this. And then the Ethnopharmocologic Search for Psychoactive Drugs made it clear that there was also some pretty interesting science around this. And so I got the idea that maybe there's a career here. Maybe I should try and become an ethnopharmacologist. And in my typical teenage way, teenage brain, I was thinking, well, actually, maybe I can actually get paid to get stoned. How cool would that be? And I managed to do it, actually. So I persisted. And time went on, and 2017 came, and we put this conference on, and we published the Symposium volume. And we reprinted the 67 book in a high-resolution, new, fancy, shiny edition, and then we also printed the 2017 proceedings, and that's what this double-volume box set is, both conferences. And even though the data in the 67 book is 50 years old, there's a lot of really interesting information there, and it gives you sort of a snapshot of, you know, what was important in the field at that time versus what was sort of now important. And if you go to the website, you can order the book or order it from ESPD50.com. And there are all the videos from the live stream are also on a website. And I'll make sure that Josh or someone has those links so you can just look at them if you like. And but the story begins earlier than that. The story actually begins in 1971 when my brother and myself and three of our equally enthusiastic friends or deluded friends or whatever decided to go to La Chorrera in the Colombian Amazonas in search of an obscure hallucinogen called ukuhe, a witoto hallucinogen made from Virola. Virola is a tree, a genus of trees in the nutmeg family, many of you know, that is used by various tribes. The sap is extracted, and the sap is full of tryptamines, DMT, 5-methoxy-DMT, and related compounds. Some tribes make a snuff out of them. Most of them make snuff out of them. But there was an orally active preparation that the Witoto used. And we were interested in that because we thought that if we could find an orally active form of DMT, naively thinking, correctly, that it would last longer. And if we could spend more time in that state, then we could learn more. When we discovered DMT in Berkeley in 67, 68, it was the most astonishing thing we'd ever encountered. You know, and we all thought, this is way beyond LSD. This is something else. This is like from another dimension or something. It's completely, you know, more outrageous. And so we basically decided, but the problem with DMT, as many of you know, it only lasts a few minutes. So it's hard to get much back other than, oh my God, what just happened to me, you know? So we wanted a more prolonged form of this, and we stumbled on a paper by Schultes in the Harvard Botanical Museum leaflets, which was called Virola as an orally active hallucinogen. I mean, just exactly what we were thinking. So we said, we read that and we said, okay, you know, we've got to quit school, quit our jobs, whatever we were doing, and go to Colombia and find this drug, ukuhe. So we set out, and eventually we ended up at La Chorrera, and this is our band. This actually, here, this is the Brotherhood of the Screaming Abyss. We kind of called ourselves that. We were serious about our work, but we have a sense of humor, and we are Irish after all. So, you know, we had this kind of tongue-in-cheek moniker for our group. And here is one of our compatriots, Sarah Hartley. She's now a psychoanalyst, a heavy hitter in Oakland. This is me. This is my brother. This is his girlfriend that he picked up along the way. And this is our friend Michael Lasky, an anthropologist, or wannabe anthropologist at the time. And we've just arrived at La Chorrera, and my brother is doing what he did so well all the time, which is roll a joint, as you could see. And at the time, cannabis was kind of loosely regulated in Colombia. So this is what led us, was the quest for ukuhe, this obscure witoto drug. But what we encountered at La Chorrera was this. mushrooms were all over the place. At La Chorrera, they had cleared maybe a couple hundred acres of pasture, and they brought in these Cebu cattle, the white humpback cattle, which happened to produce the preferred substrate for Psilocybe cubensis. And it was the rainy season. So in the fields, in the pasture, out of every cow pie were these huge clusters of the most beautiful Psilocybe cubensis we ever saw. And we had been kind of cautioned by an anthropologist on our way in that, we can't just march into the village and start asking about ukuhe. It's supposed to be a big shamanic secret, and the fact that we even knew what it was kind of, the anthropologist was not happy about this at all. And also, we were far more colorful than the Witoto. I mean, we looked like we'd stepped right out of Haight-Ashbury. So, we had beards down to our waist, we had bells, beads. We were a colorful bunch, and we just showed up in this poor man's village. No way to send a telegram or anything. He's out in the jungle. So we show up, and he's like, "You're here for what? You want what?" We said, "Yeah, Doc, and don't worry about it. We'll be cool." And so we went on, and we actually had to cross this trail from the village that he was in, called El Encanto, which means the enchanted, overland to a parallel river, the Rio. We were on the Rio Caraparaná in the Colombian Amazon, north of the Putumayo, and then we went over to La Chorrera, which was a 4-day trek along this trail that had been constructed in the early part of the 20th century to support the rubber industry. There's a whole very dark history about the rubber boom in that area. I mean, thousands, possibly hundreds of thousands of Indians were enslaved and tortured and killed if they refused to work. So there are several books written about that. One I might recommend is called The Devil's Paradise. And it recounts this essentially a Holocaust, a kind of Holocaust, where hundreds of thousands of Indians were murdered or enslaved in service of the rubber industry. But so when we got to La Chorrera, and we had kind of listened to the anthropologist, and we weren't completely clueless, so we didn't go in and start yakking about ukuhe. We decided to hang out for a while and get the lay of the land. But then these mushrooms were there, and we had no experience with them at all. We knew what they were, but we had no experience, and we didn't really take them seriously. We thought, well, these will be fun to play with while we're waiting for the real mystery to show up. Well, the mushroom quickly and we started eating them, and we started eating them regularly and in rather large quantities, in part because there wasn't all that much else to eat. So these things became kind of a regular component of our diet, and we started consuming them. And the mushrooms quickly reorganized our priorities. It became clear that they were really the real mystery. Eventually, we did find ukuhe, but when we found it, it turned out to be kind of disappointing. But the mushrooms were not disappointing, and that's how we did the whole experiment at La Chorrera and that sort of thing. And if you guys are familiar with the Terence and Dennis mythos, you can read about it in True Hallucinations or The Brotherhood of the Screaming Abyss. But suffice it to say, our expedition in some ways went completely off track, but in other ways was completely on track, because one of the consequences of going there and getting to know these mushrooms was that we brought spores back with us so that we could try to figure out how to grow them, and that turned out to be actually quite successful. So I'm not going to get into the details of the experiment at La Chorrera. You can read about that, but about 4 years after we came back, we succeeded in growing mushrooms, and we put out this little pamphlet called Psilocybin Magic Mushroom Grower's Guide under pseudonyms. We were a little paranoid. We didn't want our real names associated with it, so we called ourselves O.T.Os and O.N. Eric. Anyone know which one I am? Of course you don't. Why would you know? Well, I'm Eric, okay, and Terence was Os. And Otios means far away. The word osios means far away, and oneric means dreams. So this is dreams from far away was the implication of those pseudonyms. And it fit right in with our own delusions about the mushrooms, which was they were either extraterrestrial beings themselves or a connection to an extraterrestrial, civilization. I mean, our categories were pretty loose, right? I wouldn't call it totally airhead, but it was kind of sorry, I got my foot on the cable. Anyway, we grew these mushrooms and published this book. And partly the reason we grew them was we wanted other people to be able to grow them so that they could confirm or not the experiences that we were having at La Chorrera, because they just seemed so completely off the wall. We thought we're either nuts or you don't need. But as it turned out, they did confirm a lot of this, and now mushrooms are pretty common in the culture, one of the most easily obtained psychedelics, and because of you have to grow them so you don't have issues of purity and so on. Terence once said, we are involved in a symbiotic relationship that something has disguised itself as an alien invasion so as not to alarm us. Well, this is what we thought we were after, what we were after when we first got to La Chorrera. This is Varroa theodora, one of the species that is used to make this ukuhe. And this was part of the quest. And 10 years later, after La Chorrera, as a graduate student at the University of British Columbia, I returned, this time to Peru, to again look for ukuhe, and this time try to do some serious science, try to collect it, do some chemistry, do some pharmacology, and so on. And this time I did succeed. And interestingly about the ukuhe, it was really a dying tradition. It was not like this was actively practiced, and because of the atrocities that had been committed against the Watoto and all these related tribes in this area back in, about 1910 to 1920, it was a very culturally impacted tribe. A lot of the traditions were gone. It was, these were suffering people. They had been forcibly relocated from their ancestral home at La Chorrera to this place south of the Putumayo, up the Rio Ampayacu, but they carried their, some of their knowledge with them, and for them, ukuhe was something that they remembered their grandfathers did, or their fathers did. Most people didn't really know much about it, but we found a few who said, well, I sort of remember how to prepare it, and I'll give it a shot. I'll see if I can make it. So out of that, we got several samples of this ukuhe, which basically the sap is taken and it's boiled down to a thick paste. It's mixed with ashes, and then this is the product. You take a piece of this mass or whatever, about the size of the tip of your finger, and it's supposed to cause a very rapid and violent reaction, and you get overwhelmed very quickly. Well, we did lab assays. You guys know what lab assays are? The psychonauts among you? Lab stands for a large animal bioassay. which is mainly a human assay, right? So of all these specimens that we got, we tested every one. And only about out of 7 specimens, we couldn't detect any activity in most of them, but there were a couple that were pretty active. And then when we got it back into the lab, we could correlate that with the chemistry, and it turns out, you know, some of it turns out that Varroa is a very chemically variable species. Even the same species in different areas can have a different tryptamine profile. But that was that. I was able to work on that for my thesis, and also this is one of our informants, one of our people that was willing to try it and preparing the ukuhe. Pretty simple process. You just strip the bark off the tree. The sap turns red as it oozes out of the bark, and you heat it up and then mix it with water, concentrate it down, and you get the preparation. Well, the other half of the work I was doing in Peru at that time was focused on ayahuasca and the same considerations. By that time, a lot more was known about the composition of ayahuasca. And what my thesis was really a comparison of ukuhe and ayahuasca, because completely different botanical sources, different ethnographic context, but similar chemistry. And so the question is, we thought we knew what the oral activity of ayahuasca was about. It's about the beta-carbolines in Banisteriopsis that inhibit monoamine oxidase. So the question was, is that the same thing going on in ukuhe? Are there beta-carbolines in it? Does it inhibit MAO and that sort of thing? In other words, is it the same mechanism? But so as it turns out, ukuhe is a completely obscure, folk medicine that's pretty much disappeared, and ayahuasca's gone on to take over the world, and we're witnessing that now. You know, it's very widespread, every parts of the world. This was our informant in the ayahuasca part from Pucallpa. We went, actually this happened before we went to Iquitos to find the ukuhe, but this was Don Fidel Mosambite in front of his house. And we had his name before we went to Peru, because Terence and his wife at the time had been there in 1976 looking for ayahuasca, and they'd made contact with Don Fidel. And Don Fidel turned out to be an amazing person. I mean, not just any curandero, but a very, very good one. And not only did he know his stuff and know his plants, but he was also willing to work with, these completely clueless graduate students that showed up on his doorstep one day and basically said, "Tell us everything you know about ayahuasca." Or that's what we would have said if we had spoken Spanish, which we didn't, fortunately. But he actually got it. And he looked and he said, "Oh, yeah." So this is Don Fidel and his uncle, Don Jose. So Don Fidel and Don Jose, who worked together as ayahuasqueros in the tradition called vegetalismo, were very kind. And they let us stay at his house part of the time, drink ayahuasca. They prepared many different preparations of ayahuasca, let me take samples for analysis and all that. So he was the real deal, and not a pseudo shaman, or a real shaman. And I always wanted to get back. He was about 60 in this picture. I never was able to get back, and he passed away a few years ago. But that was the start of my other research on ayahuasca. And then eventually we collected all these plants, made samples, had a whole bunch of little bottles of alcohol, and we'd collect the samples, do herbarium specimens and so on, and eventually got these back to the university, where I spent the next 3 years sort of sorting this out. And I remember on my flight home from Lima after collecting in the jungle for 6 months, and I took a red eye from Lima to Vancouver, got in about 4 in the morning, and I had like duffel bags full of these samples and bags of dried plant samples and, you know, plant presses, press specimens and all that. Plus I looked pretty rough being in the jungle, and so I got there, and this customs agent, I said, well, you know, Miss, whatever, it was a young woman, and I said, you know, I have permits for all this. Don't worry. Would you like me to call my supervisor at UBC and confirm that, you know, I have the right to import these? And she looked at me, and she looked at all this stuff spread over the table, and she looked back at me and she said, you know, you're the kind of people my supervisor warned me about. And she said, just pack it up and get out of here. And so I did. And then I got the stuff back to the lab, and eventually actually even got a couple of papers out of it, which is sort of what you're supposed to do when you're playing the graduate student game. And so I got these 2 papers in Journal of Ethnopharmacology, which was a very new journal at that time, and that sort of gave me, I guess, a certain amount of academic credentials that led to some interesting things later on. About the same time I was doing this work at UBC, back in the lab, my friend and still very close friend, Luis Eduardo Luna, was in Iquitos doing his own ethnographic fieldwork on ayahuasca and wanting to make a documentary about it. And this is in fact his main informant, Don Emilio Andrade. And he was approaching it ethnographically, not really ethnobotanically, but he hung out with these people. And Don Emilio Andrade was a remarkable ayahuasquero as well. And he was part of a revered lineage of ayahuasqueros that you know, the descendants of a famous one called Don Salone. Don Emilio learned from Don Salone, and some of you may have heard of Don Jose Campos. He was another 2nd-generation student of Don Salone's. So it's still very much active tradition. Well, time went on, and I was doing a postdoc. After I got my degree, I was doing a postdoc in San Diego, working on something completely boring to me, not really of interest, but I was doing it, and I got a letter, or actually, yes, I got a postcard. In those days, remember, no email, none of that. People actually sent postcards around, and the custom was, if you wanted a scientist reprints, if you wanted a reprint of a paper, you sent a postcard and said, please send me a reprint. Well, one day I got a reprint request for my paper on ayahuasca from this guy here, Juan Saavedra, who was a investigator at the National Institute of Mental Health. And he requested a reprint, and as it turned out, I recognized his name because he had published a paper previously with Julius Axelrod about endogenous DMT. And he published a paper about how isolating enzymes from rabbit's lung that could make DMT endogenously. And so I knew his paper, and so I just took a chance, and I sent him the paper, and I sent him a letter, and basically the letter was, you know, thank you so much for asking for my reprint, and I'm familiar with your work, and maybe is there an opportunity for me to work with you on tryptamines? And as he wrote back, and he said, well, you know, there's this program there called the Pratt Fellowship, the Pharmacology Research Associate Traineeship. It's for people who are outside of the field to get into the field of pharmacology. You should apply. And so I did. I applied, and eventually I was accepted, and I went to NIH. I went to the Center of Biomedicine, you know, in Bethesda, Maryland, and I did a 2-year postdoc with Dr. Saavedra, who turned out to be rather difficult to work with, actually. It was kind of psychotic, but that's a whole other story. But interesting thing happened, which is that I had been there about 3 weeks. I had arrived in Bethesda, and we were in the lab, and he said, you know, there's a box of old chemicals up on the shelf here in the lab in the back. You know, some compounds that Julie and I ordered when we were doing the DMT study. There might be something interesting there. Why don't you go through this box, see if there's any interesting compounds, and send the rest of it to the you know, waste hazard facility at NIH. So I got up on the shelf, I got down this dusty box, and as I was getting it down, what do you know? Right beside it, there was a mint condition, brand new, untouched, unopened copy of the Ethnopharmocologic Search for Psychoactive Drugs. And I was just blown away. I thought, surely this is an omen of some kind. So that book just kind of quietly disappeared into my library. I went through the box of compounds, and there was some interesting stuff in there, including a couple of grams of 5-methoxy-DMT and a couple of grams of DMT and some bufotenine and a whole bunch of tryptamines that nobody's ever heard of, like 5,7-dihydroxy-dimethyltryptamine and all that. So I kept those compounds actually for many years. Didn't have the courage to try any of the exotic ones, but the others came in handy. And so, you know, I mean, it's just one of those things, you know, it's like a message from heaven or something that this book should suddenly surface. So suddenly I had 2 copies of the Ethnopharmocologic Search, one from '68, beaten up and tattered, and the other one brand new, mint condition. So I was 2 years at NIH working on this, and as it turned out, I didn't work on tryptamines. I actually worked on what was going on in Dr. Saavedra's lab was autoradiography, which is a way to map receptor sites. And we had a radioactive, one of the Shogun compounds called DOI, which was very selective for the 5-HT2A receptors, and it's halogenated. It's got an iodine on it, which is very convenient because you can put a radioactive iodine molecule on that, and then we could use it for autoradiography to map the serotonin receptors in mouses' brains, of course, not human brains. Well, so that was part of the history, and then a few years later, I was invited by the UDV, which is one of the Brazilian religions that uses ayahuasca, to a conference in Sao Paulo. And they call it Huasca. That's the Portuguese transliteration of it. And they invited myself and a bunch of other investigators in '91 to this conference. And we all came and presented our material. And then they revealed the real agenda, which was they wanted us to do a biomedical study on ayahuasca. And partly because the government was considering banning it, they weren't sure if it was, you know, drug abuse or if it was really okay or what was going on. So the UDV wanted all these foreign investigators to come look into this, figuring that the foreign investigators would bring more credibility. That's not really true, but that's what they thought. So we signed up for this study, and I was able to get some funding for it through the Hefter Institute at that time, and also Botanical Dimensions. And we came back in '93 and carried out this biomedical study on ayahuasca. And we did that in Manaus, at the Núcleo Calpari. They have temples. They have various temples spread throughout Brazil. There are about 20,000 members of the UDV in the country. And this was the oldest, this was the original temple. And in order to carry out the study, we had to make the medicine. And the UDV does this on an industrial scale, more or less, because they have to make enough ayahuasca to serve about 10,000 people every 2 weeks. So they do these massive preparos, as they call them, and you know, it's a 48-hour process many buckets going, and they harvest the vine. They grow all of this stuff. They're not ripping it off from the rainforest. They have huge cultivated plots of both the vine, Banisteriopsis, and the chacruna. So they're self-sustaining, and we prepared the material, and of course it had to be bioassayed on a regular basis over this 48-hour period, so they were sure it was the good stuff, and that was interesting. So what you're looking at is a sort of a lot of labor, a lot of work, but also a ritual, a ceremony. There's much singing of songs, and they really ritualize it when they do this. Well, finally we got the brew, and then we carried out the study. There was no place to carry it out except in the temple. So we had 15 subjects volunteered for this, and they would come to the temple every day, and we'd put catheters in their arms, and they would drink ayahuasca, and we'd take different measurements, heart rate, blood pressure, the usual kind of things that you would investigate. These are some of that's me there, and Charles Grove from UCLA, who we tapped to be the principal investigator since he's a psychiatrist. He's still active in psychedelic research. And then Jace Calloway, who is also an American, but he was at the University of Kuopio in Finland. He's a pharmacologist and a biochemist. So, and there were other, there were Brazilian and other people from other countries during this work. So we took a lot of physiological measurements, but most importantly, we did a great deal of psychological screening. We used these different questionnaires and instruments to measure things like their moods, their, mental functioning, their cognitive abilities, linguistic abilities, and that sort of thing. And the UDV members always scored slightly higher than the so-called normal controls. So there was no evidence of any impairment. In fact, they were a little more functional than their compatriots who didn't drink ayahuasca. The other component that we did that was important, we conducted these sort of psychiatrically structured life story interviews with these folks in Portuguese, with a Portuguese psychiatrist doing translation for Charlie Grobe and the others. And almost all the UDV volunteers that we talked to, their stories were similar, which was that they had come to the UDV at a time of crisis in their lives, usually very often involving alcohol or other drugs, but primarily alcohol or marital discord or, crime, all sorts of dysfunctional thing. In other words, people who were at a time of crisis in their lives. And they usually came to the UDV at a friend's invitation, and most of them reported that their initial experiences were quite terrifying. And they reported episodes where they could sort of visualize where they were going, or were shown in the visions where they were headed if they didn't change things. And, one person I remember said, I saw myself in a canoe, and the canoe was going down the river, and there were rapids, and I was terrified that the canoe was going to capsize and I'd be thrown into the water. But then I looked ahead, and there was another canoe, and there was the Mestre Gabriel, who was kind of their prophet, their Jesus figure, was standing at the head of this canoe, and I could tell then that it was all going to be okay. And so we measured all this. We published 8 peer-reviewed papers out of this study, and it's true. I mean, they all had problems, and they all felt that joining the UDV and taking ayahuasca had changed their life for the better. And if any of you have taken ayahuasca, or people come up to me all the time and say, ayahuasca changed my life, ayahuasca saved my life. Well, these people felt that it did. So that was very interesting to do that study. And that was really the first it wasn't a clinical study because it was only observational, but that was the first kind of biomedical study of ayahuasca that had been done at the time. This was 1993, and we found some key findings. There was no evidence of acute toxicity, no evidence of neurological, cognitive, or personality dysfunctions, strong evidence of positive behavioral changes, and the UDV also emphasized that it wasn't simply the medicine, it was the context, it was the very supportive environment that the UDV created. They're a very community-oriented sect or religion or whatever you want to call them. I used to kid them. I said, you guys are like Mormons. You're like psychedelic Mormons. I don't know if that means too much here in Europe, but Mormons are a kind of odd Christian cult in North America with some very strange beliefs, but they're very community-oriented, and similarly the UDV was too. And they felt that they had to be in the church to get the full benefit from the medicine, that these were not really separable. And, so we published these results in 8 peer-reviewed publications over that decade, the rest of that decade. And there was this process really started a sort of revival in ayahuasca research. So our study was done here. This is the number of publications in PubMed or Medline at the time. Well, way less than 10 right up until now. But then it started to climb. Now if you go on PubMed and put in ayahuasca as a key word, you get about 120 publications. So it's an active area of research. And in a way, this study kind of triggered that. And there's actually a fair amount of good research going on ayahuasca. Okay, I'm going to skip this. It's a little complicated. One of the interesting things that we looked at when we were doing the ayahuasca research was different admixture plants. Ayahuasca is not just 2 plants. Most of you know ayahuasca is made from the vine, which supplies the beta-carbolines, and the leaf of chacruna, or sometimes a different plant is used, but the leaves contain the DMT. But those 2 they're really at the center of a whole pharmacopoeia of plants. There's about 150 species of plants, not 150, more like 50 species of plants, 52 genera that are occasionally used with ayahuasca, either as admixtures or taken in the dietas in association with ayahuasca. Of course, in vegetalismo, in the traditional Amazonian tradition called vegetalismo, dietas are very important. You diet with different teacher plants, different medicine plants, and these are understood to be teacher plants. So you either take them with ayahuasca or you take them while you're on a long, extended period of isolation and so on. The chemistry and the pharmacology of these plants is not very well investigated at all. So what we did, this was published with Eduardo Luna and myself, we essentially did a big literature review on what was known about these plants. So we looked at 52 of these genera and whatever had been published about them, what they were used for. Interestingly, 23% of them were purported to be hallucinogenic themselves. So there's a lot, virtually unknown chemistry. 11% had unknown constituents, 6% had indole alkaloids, which are usually, that's kind of a marker for psychoactive alkaloids. 2% had other types of alkaloids, and so on. So we published this paper, and that got some attention at the time. And this is the traditional. A lot of these plants that are used in dietas are also part of just folk medicine. You can go to the herbalist stalls. There's a whole passageway at the market in Belém, in Iquitos, called the Pasaje Pascual. And this is where the herbalists sell their wares, and you can go and buy all of these mostly alcoholic tinctures, sometimes dried bark or dried leaves, but usually they make tinctures out of them. So you can go, this is like your local pharmacy. This is where people go to get their medicines. They can't afford to go to the pharmacy, you know, and actually they don't want to go to the pharmacy because they can get most of their needs met, you know, right here. So you can go and talk to the people selling the herbs, tell them what your problem is. Chances are they'll have some remedy that they can suggest to you. So then again, this is the work I did under this grant from the Stanley Foundation. I'm not going to get into details of this. This was the, at least I hope I'm not. And you know, we published, actually out of that, a grant with $500,000, we only published one paper, but it was a very extensive paper. So they got their money's worth. And just, I guess, to explain how it works, we used essentially receptor binding assays as a screen for these extracts. Receptor binding assays are pretty simple. You take a radioactively labeled molecule that binds to a receptor. You incubate it in buffer, and then you add the cold drug or the cold extract, whatever you think. Say it's a drug. For example, in this example, the drug would be domoic acid, which happens to be a neurotoxin to the glutamate receptor. You add it in increasing concentrations, and you measure what concentration is needed to knock half of your radio label off the receptors. And that piece of data that you generate is a measure of the affinity of the compound for the receptors. So it tells you basically about how potent it is, and you can screen a whole bunch of different receptors, and you can get an idea of the receptor profile of a particular, you know, of an unknown extract. So we use that. I'm not going to go into this. I'm not going to go into this. I'll go here. Some of these things did kind of rise to the surface as being interesting. Oh, thank you, thank you. One genus that sort of rose to the surface as we did all the screening was the genus Tabernae montana, which is in the Apocynaceae. It's a family that's known for its alkaloids. It's a very toxic family. And actually things like iboga, Tabernanthe iboga, is a member of the same family. And the molecules, the alkaloids in the Tabernae montanas, are actually quite similar to ibogaine and related alkaloids. They're very close in structure. And these have numerous uses in folk medicine. You know, this one is used for postpartum delivery to, you know, relieve pain and prevent hemorrhaging, used for sore throat, used for eye wounds, used to toothache. So they have many types of biological activity, as almost all these plants do. You know, it's rare that, I mean, any medicinal plant is going to be used in the context of folk medicine for a lot of different things. And this is another Tabernae montana. So of the plants that we screened in this grant, these kind of rose to the top. These 5 plants here, 2 of which were Tabernae montanas, and then from other families, they hit the receptors, they hit the target receptors. And as you see, this one here hit almost all the receptors of interest. So that's how you sort it out. So then the next step, taking this binding data, you would go and fractionate the plant and find out what in the plant are interacting with which receptors. But we never did get it that far. But that's kind of where that grant ended. But we have 6 genera that need to be looked at in much more detail. So that was the Stanley Grant. The question is, why bother to do this work? What use is the search, the ethnopharmacologic search for psychoactive drugs? Don't we have enough drugs already? Who needs yet more psychoactive drugs? Well, that's a fairly short-sighted question, because new psychoactive substances, whether natural or synthetic, structurally novel compounds often mean novel pharmacological mechanisms, novel cellular and molecular targets. So that is useful, as some of these drugs will identify a molecular target that we didn't know existed before, enhancing our understanding of the neurochemical foundations of consciousness, which we're all interested in, the neuropathology of mental illness, and eventually leads to potential therapeutic applications, potentially new drugs, and better understanding of the nature of mind. All worthy goals, I think. So here's just a quick look at some of the significant discoveries in ethnopharmacology over the last 50 years. I'm not going to go into details on this. I hope you're not completely bored yet. Are we okay? Okay. I just want to highlight some of these will be not familiar to you, and others you'll already know about. But here, for example, is one of the plants that we collected. It's called Toé negro, black toé. Toé in Quechua, toé is a local term for Brugmansia. Tree daturas are called toés. They're in the Solanaceae. They're in the nightshade family, and they are nightshades. Well, this is in the Acanthaceae, but it's also called toé, which suggests that it has similar psychoactivity, and it's called Teleostachya lanceolata. And this is not a rare plant. This is actually a plant that you can find growing along the streets in Iquitos. They grow it as a ground cover, essentially, in parks and along the traffic mediums. Its constituents are completely unknown. Sometimes it's used as an additive to ayahuasca. It's said also to be used as a hallucinogen when by itself you cook up 10 leaves for 7 hours, and then you have an intoxication that may last for 3 days, during which there is a conversation with the plant. It may also cause you to go blind for 3 days. So, very interesting plant. I have never bioassayed it, and I have no intention of bioassaying it. That's what graduate students are for. Here's another one that you must know about. This Vinho de Urema is Mimosa hostilis, and in northeastern Brazil, this is the source of an orally active DMT preparation called Urema. Well, we know it contains DMT, rather high levels. In fact, often this is an alternative, kind of a commercial alternative to the traditional admixture plants. Mimosa hostilis is a great source of DMT because you can easily isolate grams and grams of it out of the roots. But it's not understood why it's orally active, right? DMT should not be orally active, and there's no beta-carbolines or other plants added to it. Well, recently a new compound emerged or was discovered called urimamine, and it may be that this is the answer. You can see that this contains DMT kind of incorporated into the molecule, and it may be that this is an MAO inhibitor, or it may also be, maybe it's psychedelic in itself. So far, not enough material has been isolated in a pure form to bioassay and determine whether that's true. But this is another example of ethnopharmacology, novel molecule that may have some interesting properties. Another thing that was not even on the radar at the time we did our work were the acacia species. Acacias, particularly the Australian acacias, are some of the highest level, most potent sources of DMT and other tryptamines in nature. This is a particularly rare one, Acacia phlebophila, and the leaves of this plant can contain 2% DMT, which is very high. And there are 1,700 species of acacia all over the world. So there are many, many acacias that contain tryptamines. Interestingly enough, these acacias are mostly indigenous to Australia. They're called wattles, which is the national tree of Australia. But Australia has very draconian drug laws, so they've actually just declared all wattles illegal. So the national tree of Australia is now a Schedule I controlled substance, which doesn't make any sense, but drug laws rarely do. Interestingly, the DMT Nexus on the web, if you know this website, if you want, this is like your go-to site on the web if you want to know about tryptamines. That's its focus, is everything about tryptamines, chemistry, pharmacology, ethnobotany. So it's worth taking a look at. Here, of course, is the South American toad shamanism, Bufo marinus and Bufo alvarius. Bufo alvarius, source of the famous toad medicine that Octavio works with, contains 5-methoxy-DMT in its venom glands. Most Bufo species contain another compound called bufotenine, which is actually a toxic compound. It's a pressuramine. It's very hard on your blood pressure. So something about this species, probably a single gene mutation somewhere along the way, caused Bufo alvarius to make 5-MeO-DMT instead of bufotenine. But as Octavio mentioned in our conversation, he said, well, because we were talking about the apparent, the archaeological evidence for toads being used even in the Middle East, even in other parts of the world. And I said, but what about the restriction of it? And basically the answer is maybe there are other species of Bufo that contain 5-MeO-DMT. This is the only one known so far, and its range is very limited. Isn't that right, Octavio? It's not found many places. In the Sonoran Desert, essentially.
The Sonoran Desert is a 200,000 square kilometer area. So
Yeah.
It's large. Like you need to cross maybe 10 hours driving non-stop to cross whole Sonoran Desert. So it's big.
Yeah.
And because it's big and there is large amounts of toads, it is possible that this medicine could travel and be spreaded all over at least the Americas. And because there is gold from Colombia in Canada, and there were these trade routes from all goods, and people were not exchanging only letters or feathers. They were also exchanging this kind of really powerful tools.
Yeah, there needs to be a lot more field work done with this, and this is a whole very fascinating area that's just opening up. And I hope by the time we have ESPD50, if I'm still alive, Octavio will be able to come and give us the full story, right? So there's that. Then this is an very interesting plant. This is Salvia divinorum, and the compound salvinorin A. And Salvia divinorum is a mint. It's a member of the mint family, and it's used by the Mazatecs as a shamanic medicine. And it's interesting for a number of reasons. For one thing, the compound, salvinorin A, which is the main compound, doesn't contain nitrogen. It's not an alkaloid. That's very rare for a CNS active compound. Most of these things have nitrogen in there somewhere. They're basically alkaloids. Another example of psychoactives that are not alkaloids are cannabinoids. But this is very interesting. It's actually the most potent psychoactive substance yet discovered. It's active at around 50 micrograms. So it is as potent as LSD, but certainly no resemblance to the structure of LSD. And and it doesn't work on serotonin receptors like most psychedelics. It actually works on kappa opiate receptors. The there are three main kinds of opiate receptors: delta, mu, and kappa. And salvinorin A is absolutely selective for the kappa receptors and no other receptors. This is unheard of in pharmacology. These are different receptor subtypes down here: serotonin, dopamine, beta receptors, opiate receptors, and so on. The red blocks, this is LSD, and this is more typical of a drug. Most drugs are dirty drugs in the sense that they don't have that much selectivity for receptors. And you know, the LSD hits mainly serotonin receptors, but it hits a bunch of other things too. Salvinorin A hits only one receptor, which is really, and it hits it very hard. It's quite potent. So that's pretty remarkable. And actually Brian Roth, who was in the movie, is the guy that worked out all this pharmacology with salvinorin A. And he told me, you know, if he had wanted to design a drug that would be selective for the kappa receptors, he said it would not look anything like salvinorin A. He said the stuff looks like cholesterol. You know, I can't believe that it has this kind of activity. So again, ethnopharmacology trumps computer-aided drug design. I'm sorry I had to use that word, but anyway. So this is a contribution of ethnopharmacology that's worth talking about. This is kava. Many of you know kava. It's a plant in the pepper family, Piper methysticum, used throughout Polynesia as the source of a relaxing, kind of a relaxing beverage. And it's, you know, it's benign. It's, you know, if you go to Polynesia, instead of being served coffee, you'll be served kava. And it's a very important drug for like socialization. It makes you feel quite calm and relaxed, but you don't lose judgment and you don't lose your ability to talk. So it's really a social lubricant in some ways. And it's got a lot of beneficial properties and a long tradition really throughout Polynesia and parts of Micronesia. It's used, and the roots are what is used, and these are the compounds, the so-called kava lactones, and these also don't contain nitrogen, so these are not alkaloids. Again, no nitrogen in those things. Some of these are sedatives and muscle relaxants, and others are euphoriants. And there are many strains of kava, many different chemotypes, and each chemotype has a unique psychopharmacological profile. Sometimes they give it to you if you want to sleep, they'll give you one type. If you want to just feel good and happy, they'll give you another kind. And there are many kinds. It's kind of like cannabis, you know, they're distinguished based on their pharmacological properties rather than their appearance. And they're good anxiolytics, they're good for depression, they're a good muscle relaxant, good for sleep. They're anticonvulsant. They can be used to treat epilepsy. They're neuroprotective. They can protect the brain in cases of stroke. They're an analgesic, non-opioid analgesic. They're a local anesthetic. If you drink kava, your tongue and lips and mouth will get numb immediately if it's properly made. They're also very good topical antifungals for some reason. So kava is one of these things. It was actually discussed in the '67 book, and there's been quite a lot of work on it since, and it's been accepted. It's not illegal. You can buy it as a dietary supplement in any health food store. Iboga, we know about. Iboga is another member of the Apocynaceae, and it's used to treat addictions very effectively. It was known about in '67, but it was not included in the symposium. But now there is a lot of interest in ibogaine, which is effective for treating addictions, especially opiate addictions. It is a psychedelic. It has profound psychedelic effects, but it's also kind of unique because it blocks craving for opiates. So addicts can take it, they have a revelatory experience where they kind of see how to get their lives back on track, but then it relieves the craving for opiates, sometimes for a long time, sometimes for a few days, a few weeks. So if the people have their act together, they can use that time to kind of make the behavioral changes that they need to make in order to make the therapy effective. If not, they'll just fall right back into it. But this is an interesting plant, and you know, it has numerous neurotransmitters interaction. It's not only serotonin, dopamine, GABA, muscarinic receptors, and so on. So it's not a true psychedelic in the sense that it's not one of these 5-HT2A agonists that the classical psychedelics are. Yeah, so it's used to treat addictions primarily, and now with the opiate epidemic going on, you'd think there would be steps being taken to, you know, to get it approved to treat that, but that's not the way it works. Another interesting plant, this one from Southeast Asia, is kratom. You've probably all heard of this, Mitragyna speciosa. It's in the coffee family, the Rubiaceae. And in Thailand, other parts of Southeast Asia, it's used essentially as an opium substitute. And the alkaloids in kratom, mitragynine and 7-hydroxymitragynine primarily, are potent mu-opiate agonists. So they hit the same opiate receptors that morphine or heroin or any of the classical opiates hit. But potentially, it's less toxic, and it may be part of the answer because it could be a kind of herbal methadone. They often give methadone to addicts to help them, you know, it essentially replaces the heroin. It's not so psychoactive. This could potentially be used for that. And in fact, in Southeast Asia, many people take kratom if they're addicted to opium or addicted to heroin. It helps them taper off and discontinue it. So overall, it's beneficial. But of course, it's been identified as a major threat, which it's not. But anyway, here's another interesting plant. All of these, the reason I'm sort of highlighting these is most of these are in the second volume of the book. This is Scaletium tortuosum from South Africa, the source of Kana. And it the roots are chewed or inhaled as a snuff, and it's a mood elevator, it's euphoriant, it's anxiolytic, it's analgesic, it's an appetite suppressant, and many of the alkaloids are serotonin uptake inhibitors, so they're just like SSRIs. But there are a lot of, some of the alkaloids are not, you know, have not really been looked at for that. Serotonin uptake inhibition is only part of the picture here. And this also has been developed as a natural health natural health product in Canada and also in the States. You can buy the extract of this under the name Zembrin and use it as a mood stabilizer and an anxiolytic. And again, it's kind of an example of what sometimes happens to these traditional medicines. They don't go the route of being developed into pharmaceuticals. Instead, they end up as dietary supplements or, you know, herbal medicines. That's the regulatory framework. And that's probably a good thing because it costs about $1 billion for each pharmaceutical that you might develop from a natural product. And in the, you know, in the extract form, Zembrin works very well. And again, this is discussed in detail in that book. We also know, besides the bufo species, there's Philomedusa bicolor, which is the source of the Cambo medicine. And here, the active principles are peptides of various kinds. And, you know, have many different types of activities, hypotensive, tachykinin, they have behavioral responses, smooth muscle contractants, arterial vasodilators, and opiate-like activity. And you know, this frog has been known for a long time, but the chemistry is complex and is still being sorted out. But potentially this may yield new medicines as well. Well, so that's 67 to 2017. What else is out there? Is there a new frontier for drug discovery and ethnopharmacology? Well, maybe. Interesting rumors have surfaced about some Boletus species in China and actually in New Guinea as well. And Boletus is not normally known as a source of psychoactive mushrooms. It's a source of delicious edible mushrooms. But there are rumors about some species which often stain blue, which is a kind of often a tracer sign of psilocybin in psilocybes. But there are these kinds of mushrooms called jiao ren ren, the little people, or the little person mushroom. And when you take jiao ren ren, it's not invariably psychoactive. It's sometimes psychoactive, and it has to do with either it's undercooked or it's not cooked enough or it's overcooked, it's not clear. But when it is psychoactive, you get these very strange hallucinations where the little people come out. And one description was a woman spent many hours looking at the drips from her faucet in the sink, and the drips were coming out, and they would, as they would drip down, they would transform into insects and fly away. And she was quite amused by this, right? So there's unknown chemistry here, and this is a very interesting thing, but hard to nail down. I had a friend who got all excited about these mushrooms, and he spoke Chinese and spent a lot of time in China. So he went to Yunnan during the mushroom season, went to the market, got every kind of Boletus that he could get his hands on, and he and his friend ate Boletus until it was coming out of their ears. They couldn't get any effect. They could not replicate it. But then I've heard other reports where, you know, it is. So there's, again, it's probably not psilocybin. It's some much more unstable compound. Maybe a tryptamine, maybe not. Also, there are rumors of psychotropic fish and actually psychedelic fish. There are a few examples of them, and they go by different names, like the dream fish, because it causes dreams. There's one species native to Hawaii that's called the dream fish, and very often the hallucinations that it produces are quite bizarre. There was one species I just read about recently, and it's not on here because this is an older slide, but very colorful fish, just amazingly beautiful rainbow colored fish. The fish looks like a psychedelic experience, and apparently if you consume it, and it used to be consumed by the Roman troops, so it was as a recreational drug, and it causes an intoxication that can last anywhere from a day to a couple of days. And people have described, one fellow had visions of giant tarantulas that were attacking his Volkswagen as he was trying to drive home after one of these sessions. Doesn't sound like a recreational drug that I'd want to take, but you can't, there's no accounting for taste. But this is interesting that in fish, and again, these compounds are ephemeral. They don't invariably show up. They're a seasonal thing, which indicates that it's probably not the fish. It's something that grows inside the fish, something the fish are eating, maybe an algae or a dinoflagellate or something like that, that actually produces the toxin. So this is an area of intense interest. What about insects? Insects have, as we talked about yesterday, these complex chemical relationships with plants. Are there any known psychoactive insects? Well, it's again, the answer is probably there are quite a few, but there's no work done. So, you're not going to find it if you don't look for it. There is one indication that there's, well, I found a paper called A Pointer to a New Hallucinogen of Insect Origin, it's called. And it's actually the larva of a moth that incubates in common bamboo and in the Brazilian province of Minas Gerais. And when the intestinal parts of the larva are eaten, it induces fantastic dreams lasting more than a day. So again, we know it works, people have experienced it, nobody knows what the chemistry is or the pharmacology of this psychoactive moth. So, you know, there's enough work to keep a whole generation of ethnopharmacologists busy for a long time. Another area that I'm quite interested in are fungal endophytes. Endophytes are fungi that grow inside of plants, and they're not pathogens, they're symbiotes. They grow in the plants, and in China, for example, there's a grass called drunken horse grass, and Achnitherium inebriums, and it has an endophyte, the endophytic fungus. Well, it makes all of these different kinds of lysergic acid derivatives, and fungi often do in you know, related fungi. So we know the structures of these. Whether any of them are psychoactive is not clear, but people are you know, looking into it. They're a good source of ergot alkaloids, and because ergot alkaloids are restricted, because you can make LSD out of them, so you have to, you know, you have to have permits to investigate this fungus. But that's just one example. There are hundreds of species of endophyte, and virtually all plants have endophytic fungi. So one of the long-term plans I would like to do, if I ever get funding, is to create a culture bank of fungal endophytes from the Amazon, and then just have those for chemical screening and drug discovery. So that's a quick look at the past, present, and future of ethnopharmacology. Thank you.